Next this afternoon, we have Thomas Meaney from Cell Free Technologies. Thomas.
I love technology! I started my career as a physicist, building semiconductor sensors like this one. One day my boss said to me, we need to align ourselves with Toshiba's strategic healthcare vision. And he asked, what can we measure? The cell, the incredible microfactory with everything pre-programmed for life. But there was a problem. In order to produce our product, we needed a biosafety lab constituting a capital investment of just shy of $1 million. And for every chemical we wanted to use, and there were hundreds, we needed to review and summarize a 10 to 20 page material safety data sheet. For example, for dihydrogen oxide, it ran to 7 pages. The common name is water. So I quit that job. Meanwhile, on the other side of the country, my co-founder, Ian McDermott, was grappling with challenges of his own. Ian is an expert protein engineer working in the University of Manchester, and he was developing high-value pharmaceuticals from cells. He was facing the challenge of low yield and long time to manufacture. Over our shared war stories, it was love at first sight. So our solution is a technology that involves breaking open cells very delicately and preserving the biomolecular machinery and extract that is responsible for reading DNA and writing protein, enzyme, and molecule. Most importantly, this is no longer classified as an organism, and therefore we're free from any biological hazard regulations or genetically modified organism regulations. So you can make insulin or other drugs, not in a factory, but right where they're needed. Not only that, but we can prototype protein in hours, not days. This allows us to produce an extract that empowers you to create almost anything. All we do is we take a piece of DNA, such as this, we add it to a tube of our extract, and in this case, it's programmed to produce green fluorescent protein. Now, this isn't the incredible part. What's impressive is that we were able to produce this extract for 10 times lower cost than before we entered the RebelBio program through our scaling. Now, that's a commercial game changer, and it made my co-founder Ian very happy. So we do have our first product, and our limited alpha release is already 4 times oversubscribed. Our core alpha testers are teachers, students, creatives, and scientists from the universities of Cambridge, Toronto, and the Royal College of Art. Our paid beta launches in September and moves to direct sales from our platform, a US biotech marketplace called the Odin, as well as package sales with collaborators building hardware and software in the field. We're shifting gears and approaching scale production. We need to secure our great team with salaries. We need to shift from small batch to large reactor. We need to reach every user on the planet. As we are a high potential growth startup based in Ireland, your investment will be joined by an optional 25K from our lead investor, SOS Ventures, as well as a further optional 250K from Enterprise Ireland. We are Cell Free Technology, and we enable inspiration.
Incredibly innovative, isn't it? In terms of suddenly taking the machinery of the cell and getting it to do other things that you couldn't do before.
Yeah, I mean, it's a first. I think we were discussing it earlier as the Raspberry Pi of biology.
Yeah, so John was saying earlier that like biotech now is where computers were in the 1970s, and this is a similar thing, isn't it? Is people will be able to program the cells themselves. Is that the idea?
Exactly. I mean, what we'd love to see is that this technology is what the 17-year-old in his basement starts using in 5 years' time in order to program the next biological application.
And it's using the DNA as turning it into a language, turning it into the programming language.
Exactly. I mean, this is already the oldest language in the world, right?
Yeah.
So it's the natural thing that we should have been doing centuries, millennia ago, billions of years ago.
I remember reading scientists saying that one day we'll use all of plant DNA and stuff as kind of the way that we use data storage facilities today. And so you can imagine going up and like flashing your phone on a leaf and it's stored all of Netflix or something on it. I mean, DNA can just do everything for us, right? Is that a realistic future? That's the one I'm thinking about.
It's not outrageous. I mean, you know, in a single teaspoon, I think they estimate that you can store, you know, exabytes. I think is the term. Exabytes of data, sufficient amount of data to cover all of human history. So I mean, that's extraordinary. And you know, when you have that capability in, you know, your foundation of your technology, I think what you can build upon that is you know, That's the part that gets
I
me excited
I
anyway.
think it was only last week or the week before someone took a GIF of a horse and programmed it into an E. coli DNA. And not only that, the E. coli then had babies almost, and they still had the GIF inside them.
Wow.
That's just unbelievable.
Incredible.
Yeah,
And that's
it's
happening now, and we're talking about the future, right?
Well, exactly. What can happen in 2 years' time?
Amazing.
Fantastic, Thomas. Thank you very much indeed.
Thank you,
Brilliant.
guys.